Chapter 13, Molecular Imaging of the Brain with Positron Emission Tomography. The objectives of this chapter are to first review the major principles and applications of positron emission tomography, or PET imaging of the brain. PET imaging consists of 5 major steps. The first is production of positron-emitting nuclei using a cyclotron. The second stage involves radiochemical procedures to attach this positron-emitting nuclei to a molecule of interest, for example, a tracer or a probe. The third step is to measure the radioactivity through use of incident detection of gamma photons on a tomograph camera. The fourth step is to take this information and use mathematical image reconstruction techniques in order to create the image. The fifth step is to apply this in either a dynamic or static sense and apply tracer kinetic models to interpret temporal and regional changes in the tracer uptake. PET imaging is a powerful technology for non-invasive imaging of molecular and biologic processes within the central nervous system. And this chapter will review the major principles and applications of PET imaging within the brain. PET imaging has a number of applications. PET imaging can be used to look at gene expression, specific enzyme activity, the pH or acidity of the environment, glucose metabolism, blood flow, blood-brain barrier permeability, neurotransmitter reception and characterization of this receptor, drug pharmacodynamics. It can be used to evaluate brain function within the motor system, the visual system, the somatosensory system, and even a variety of cognitive networks. It can also be used to look at neurochemical alterations in a variety of pathologies, including movement disorders, dementia, psychiatric disorders, epilepsy, stroke, brain tumors, traumatic brain injury, and chronic pain syndromes. The first step in the process of PET imaging consists of production of the positron-emitting nuclei within a cyclotron. To create radiopharmaceuticals required for PET imaging, positron-emitting isotopes are first generated in a cyclotron. The cyclotron is a machine in which charged particles, like protons, are accelerated along a circular path to achieve high kinetic energy. These particles are then directed out of the cyclotron via a stripper foil, where the beam is guided by a number of bending magnets. The accelerated proton beam is then often guided through another set of linear accelerators until it bombards a stable target, such as oxygen-18 or nitrogen-14, depending on the isotope of interest. This interaction results in a new, highly unstable, proton-rich isotope that becomes more stable through radioactive decay, a process in which the proton is converted to a neutron, a positron, which is antimatter and equivalent to a positively charged electron, and a neutrino. The positron and neutrino are emitted from the nucleus of this radioactive species at a decay rate that's characteristic of the particular isotope. Common positron-emitting isotopes often used for PET imaging include different forms of oxygen, nitrogen, fluorine, such as fluorine-18, and carbon, such as carbon-11. But other isotopes are also used, including rubidium, gallium, copper, chromium, and indium. A series of chemical purification processes then occurs following generation of these radionuclides, followed by radiochemical procedures to incorporate the radioactive tracers into a wide number of molecules, including physiologic substrates like glucose or oxygen or amino acids. And because these compounds are synthesized in very small quantities, really in nanomolar concentrations, they're often referred to as tracers or probes. The end product is then injected into the body, usually intravenously, before or during PET imaging acquisitions. The next step in the process of PET imaging consists of radiochemical procedures to attach the positron-emitting nuclei to a molecule of interest, creating the tracer or the probe. This slide shows an example of how to synthesize FDG. You start with an aqueous solution of fluorine-18 combined with potassium carbonate along with cryptofix 222 and through a series of nucleophilic substitution and chemical processes, the end result is a fluorinated atom attached to a deoxyglucose molecule that can then be injected into the body. Once the tracer has been created, the next step involves measurement of radioactivity through incidence detection of gamma photons on a tomograph camera. During and following injection of the PET tracer, it continues radioactive decay, emitting positrons and neutrinos. The neutrino passes through the body and is otherwise undetected. The positron travels a short distance, approximately about 1 millimeter, before colliding with nearby electrons, resulting in annihilation of the two particles and conversion of their mass into two gamma-ray photons, each with an energy of 511 kiloelectron volts and projected at approximately 180 degrees from each other. A pair of detectors set at 180 degrees apart from each other then registers the time of arrival of these incident photons, creating a line of travel or a coincidence line. For a given region having a high concentration of the particular PET probe, photon pairs are emitted in all directions and detected by a circumferential ring of scintillation detectors. This composes the gamma camera or tomograph that then converts these high energy photons into electrical signals. When the coincidence lines from many different angles are combined, the resulting data are reconstructed through mathematical algorithms or reconstruction algorithms to establish the location of each emission and form the resulting PET images. There are a number of technical parameters that influence the image quality on PET scans. The first parameter is sensitivity. This is the fraction of positron emitting events that are detected by the scanner within a particular field of view, or FOV. The second is the spatial resolution. This is the smallest distance between 2 points that can be reliably distinguished by the scanner, or the measure of the ability to accurately localize different regions of interest. The full width half maximum, or FWHM, is the width of the point spread function, or response of the imaging system to a point source of radioactivity, and this is really used to define the spatial resolution and physical limits of the device. Partial volume describes errors that result from activity in regions that are less than 2 times the full width half max. This can lead to over or underestimation of the true activity. Lastly, image reconstruction, or the mathematical process of creating an image from the series of gamma photons, also leads to differences in image quality. The next step of the process of PET imaging involves mathematical image reconstruction. There are a variety of mathematical algorithms to use for image reconstruction in PET. The oldest, most widely used technique is filtered back projection. It involves back projecting the event on an image over the line of coincidence, connecting 2 photon events, then using a convolution filter to remove any blurring. The maximum likelihood expectation maximization, or MLEM, technique is an iterative reconstruction technique where back projection is first conducted. Then a projection image is decomposed from the image guess. The differences are compared between the guess and the actual image. Then this difference is back propagated in a feedback or iterative process to further improve image quality. Ordered subset expectation maximization, or OSEM, is similar to MLEM, only it uses a subset of the projection angles per iteration, and it's more efficient. There are also a number of adaptive or Bayesian or AI or machine learning based algorithms, and these are advanced methods of using mutual information from CT or MRI to constrain the reconstruction. Or there are ways of training the algorithm to go directly from the coincidence line or the projection space directly into image space without the mathematical operators. Once the images are reconstructed, tracer kinetic models are used to describe the distribution of PET tracer throughout the brain and the body on the basis of the tracer's delivery, uptake, and metabolic characteristics given a set of serial or dynamic imaging data. Tracer kinetic models provide additional information beyond merely static images and are based on the compartmentalization of the tracer and exchange between different various compartments. The resulting images are created after fitting PET data to a compartmental models on a voxel by voxel basis or a region of interest basis. And these are often referred to as parametric images. They reflect images that show a particular pharmacokinetic parameter. This slide shows some examples of various pharmacokinetic models that are used for different PET tracers. One of the simplest kinetic models used in PET imaging is the 3-compartment tracer model for FDG PET. After intravenous injection of the tracer, the time course of the tracer concentration in the plasma from blood samples is typically quantified, assuming that there's little loss in the tracer and it only disperses throughout the body. We can look at an arterial source or a plasma time curve, and this is often referred to as the arterial input function, and this can be used to provide a quantitative estimate of the tracer availability to the brain compartment as a function of time. For this model, the FDG exchanges with the extracellular, extravascular space through the blood-brain barrier by the forward rate constant K1 and a backward rate constant K2. Both glucose and FDG are transported by the same carrier across the blood-brain barrier, so both FDG and glucose transfer are assumed to be similar, and both compounds enter the glycolysis cycle and are phosphorylated by hexokinase, or HK, which converts glucose to glucose 6-phosphate. However, FDG 6-phosphate is not a substrate for further metabolism and it's trapped within the cell. Accordingly, the FDG 6-phosphate accumulates in the tissue in proportion to glucose metabolic rate of the tissue. On the basis of this 3-compartment model, as shown in the figure, the cerebral metabolic rate of glucose, or CMR-GLC, can be calculated as the plasma concentration divided by LC multiplied by K1 multiplied by K3 divided by the quantity K2 plus K3. PET imaging has been used in a number of movement disorders. For example, Huntington disease, a tracer known as carbon-11 SCH23390 was designed to look at the D1 receptor, and carbon-11 raclopride was used to look at the D2 receptor, dopamine receptor binding, showing a reduction in dopamine binding within the striatum related to loss of dopaminergic cells. In Huntington disease as well, FDG PET reduction was also identified as part of this general degenerative process. Some other tracers with novel targets like phosphodiesterase 10 have been imaged using carbon-11 IMA 107 or carbon-11 PBR28. Additionally, PET imaging has been used to look at Parkinsonian syndromes. F-dopa PET, for example, has been used and shown that there is a reduction associated with dysfunction in dopaminergic terminal activity in Parkinsonian patients, as you can see in this figure. Movement disorders, including essential tremor, have also been investigated using PET imaging. However, there have been no known or identified F-dopa abnormalities, but blood flow has been shown to increase in the cerebellum, and there has been shown to be isolated uptake of FDG within the medulla and the thalamus. In carbon-11 flumanzyl, PET shows reduced GABAergic function in patients with essential tremor. In Alzheimer's disease, bilateral temporal-parietal hypometabolism has been identified in FDG PET and has been shown to have more than a 90% sensitivity and more than a 70% specificity for Alzheimer's disease. And this becomes more widespread with increased disease progression. It does not seem to be generalized to subcortical structures, and FDG PET has been shown to be useful for evaluating prognosis and the effects of any interventional therapies. In contrast with Alzheimer's disease, Lewy body dementia shows hypometabolism on FDG PET that extends into the primary and secondary visual cortices, whereas frontotemporal dementias show hypometabolism extending to the frontal lobes. In psychiatric diseases, dysregulation of the dopaminergic system has been shown in patients with psychosis or people at high risk for psychosis. Positive symptoms in schizophrenia appear to be related to increase in F-dopa uptake. F-dopa is also decreased in major depression and bipolar disorders. Major depression specifically has shown a reduction in the D1 dopamine receptor binding using carbon-11 SCH23390. Psychotic patients with bipolar disorder show higher D2 receptor density within the caudate, and non-psychotic patients do not appear to have this same variation in D2, and they appear to be pretty similar to healthy participants. There has been noted higher serotonergic transporter binding in patients with bipolar disorders, as measured with a variety of tracers. There's no generalized FDG uptake differences in schizophrenia patients. However, patients with obsessive compulsive disorder show increased glucose uptake in the singular cortex, thalamus, and putamen. OCD patients with lower pretreatment FDG PET have a better response to a variety of therapies, whereas patients with higher FDG uptake who have initial decrease do better on other types of therapies, particularly if they have abnormalities within the frontal lobe. In epilepsy and other seizure disorders, hypometabolism on FDG PET has been shown to correlate strongly with the epileptogenic foci. FDG PET also shows focal abnormalities in otherwise structurally normal brain, both in ictal and interictal phases. During partial seizures, however, ictal glucose metabolism and blood flow increase with epileptogenic focus, and hypermetabolism can remain elevated for 24 to 48 hours. FDG PET is very important in temporal lobe epilepsy, as favorable clinical outcomes following surgical intervention drive the need for more minimally invasive surgeries, and thus using image-based decision-making. Studies estimate that more than 90% specificity and 80% sensitivity for identifying the seizure focus in temporal lobe epilepsy using FDG PET. Carbon-11 FMZ, a GABA antagonist, shows similar co-localization in seizures involving GABA inhibitory mechanisms, and shows smaller regions in size compared to those hypometabolic areas in FDG PET. Carbon-11 FMZ was also associated with earlier age of seizure onset and longer duration of epilepsy. However, the accuracy for identifying the lesion foci is only around 80%. FDG PET has been used extensively in studying brain tumors, and historically has been shown to be useful for a wide range of applications, including diagnosis, prognosis, detection of tumor recurrence, tumor grading, and as a tool for looking at response to radiotherapy, chemotherapies, and other agents, including anti-angiogenic treatments. FDG PET has a variety of limitations because the background uptake of normal brain, particularly in the cortex, is very high, and because stromal or inflammatory cell metabolism may also have elevated FDG uptake. Amino acid PET is favored because of better specificity for tumor and less background uptake by normal tumor. This includes C11 methionine, C11 tyrosine, FET-PET, and F-DOPA as analogs for methionine, tyrosine, and phenylalanine, respectively. Other tracers like FLT can be used for looking at proliferation, and F-mysil for hypoxia. These have shown some value in quantifying tumor response to a variety of therapies, but are a little bit less available. Following traumatic brain injury, a cascade of secondary injury mechanisms follow the initial primary insult. Among these changes are alterations in both blood flow and glucose utilization. During acute injury, there's uncoupling between blood flow and glucose metabolism, as been quantified by FDG PET. Hypermetabolism on FDG PET is also common in the acute setting, and alterations in the chronic stages have been reported. However, both hyper and hypometabolism have been reported to be linked with outcomes. In migraines, hypermetabolism on FDG PET has been observed, primarily along the vestibulo-thalamical cortical pathway, in particular in spontaneous migraines. Hypometabolism has also been noted on FDG PET and has been observed in the visual cortex, bilateral insula, anterior cingulate cortex, posterior parietal cortex, and premotor, prefrontal, and primary somatosensory cortex in migraine patients at rest. Perfusion, as measured with radiolabeled water, appears lower in specific brain regions during headache, but oxygen metabolism and extraction do not appear to be altered in patients with migraine. There have been very few studies using PET imaging in chronic pain. Radio-labeled carbon-11 carfentanil has been used to quantify opioid receptor binding and has shown a reduction in the mu-opioid receptor binding within the amygdala, cingulate, nucleus accumbens, specifically in patients with fibromyalgia, and reduced binding bilaterally across the brain in peripheral neuropathic pain and localized reduction in central neuropathic pain, suggesting that opioid receptors may be altered in patients with different chronic pain syndromes. In summary, PET imaging remains a useful, flexible, and non-invasive technique for understanding the biochemical and molecular alterations in CNS pathologies. PET studies that use radio-labeled carbon-11 usually help label drugs to better understand their pharmacokinetics and pharmacodynamics. PET studies that use fluorine labels are typically used for biological compounds, and this might include glucose and neurotransmitter or ligand binding characteristics. PET studies using isotopes with longer half-lives, like copper-64, are often used for labeling antibodies and other things that stick around in the body for a long period of time. And continued development and testing of targeted PET tracers will allow molecular imaging to stay relevant for neurosurgical and neurological disease diagnosis and management well into the future.